And the title is Automated Detection and Tracking of Many Cells by Using 4D Live Cell Imaging Data.
Thank you so much. First of all, I would like to say this is my first time to come ISMB, so I'm very glad to come here. I will show a series of image processing techniques for detecting and tracking many cells in 4-dimensional live cell imaging data. This research is motivated by an ongoing project on neuroscience using C. elegans. C. elegans is known to have approximately 300 neurons, and the synaptic connectivity of the circuit have been completely identified, but its working principle is poorly understood. We are now developing a novel live cell imaging techniques to monitor the activity of all neurons of C. elegans in real time. So, for a given stimulation, we will be observe how the input signals are processed in nervous system of animals. Okay? So let's see our data. Modern live cell imaging techniques allow us to visualize more than 100 neurons simultaneously in the living state. Our data are characterized as follows. The globular object in this image corresponds to each neurons, and basically they are ellipsoidal, but some of them are slightly deformed ellipsoidal. And sizes and shapes of neurons are very similar, so it is difficult to distinguish them based on the size and shape. Such globular objects are distributed densely in a 3D space. And during the measurement, C. elegans are fixed at a plastic microtube, but they can slightly change their body posture, so accordingly, cells move rapidly and coordinately. The aim of this study is to present automated procedure for detecting and tracking many moving objects to quantify the neural activity of each neurons. So in this paper, we present automatic method, automatic detection and tracking method based on the kernel density estimation. Okay. This is an outline of the proposed methods. Our method consists of 4 internal steps: conversion, detection, tracking, and segmentation. These 3 steps are addressed based on the continuous function obtained by a kernel density estimation. The kernel density estimation is the method for estimating the continuous probability density function from digital image. We are now given a set of voxel positions in a 3D space, denoted by X, and this is a normalized intensity W, which is assigned to each voxel. So we obtain the kernel density function P of X as a combination, as a mixture of Gaussian distribution. And the mixing rate is the normalized intensity, and the Gaussian distribution is centered at the position of each voxel, like this. So in this way, we obtain the kernel density function in continuous space. Here, we make an assumption. We assume each cell is present around the local mode of this density function, where the gradient is exactly 0. Okay? So then the tracking is addressed by tracking of the local mode of the density function, which are changing over time. At the initial position, we can identify the position of cells by finding this local mode of the density function, and the number of cells are fixed here. The important point is here that we have a continuous function Okay, so these local modes can be found using well-established optimization techniques designed for a continuous function, such as gradient descent algorithm. This is the fundamental idea of the proposed method. Our paper shows a hill climbing method for detecting local mode local mode of a continuous function in a 3D space without any knowledge. Technical details are omitted here, but the idea is very simple. The process of hill climbing starts from a random position in a 3D space, and each process monotonically climbs a hill following this recurrent formula, which is derived upon an idea of EM algorithm. And each process is theoretically ensured to converge at the nearest hill. So if this method works well, all local modes of the density function can be found like this. But here, we face the difficulty. In the case of independent search, many processes starting from different initial points will converge to the same local mode, possibly which correspond to the large and bright cell. So the identification of all existing local modes is very difficult, especially for dark and small cells. To overcome the difficulty, we extend the hill climbing method to work in parallel, called repulsive parallel hill climbing method. This method works with multiple local optimization process, and the repulsion force is acted when the different process get close. Okay? In this way, a different process explore a different local mode, and we can detect much more diverse objects with just one-time parallel run. And this is the definition of repulsion force. Suppose process A and B are now climbing the same hill, then we remove the kernel density function around A from the updating process of the current position of B. In this way, the kernel density function for process B is shielded. Accordingly, process B moves towards a different hill, like this. So, I'd like to show a demonstration movie. 45 objects are included in this image, and yellow dots indicates initial point. And now 80 initial points are placed in the corner of a 3D space around here. So in the case of no repulsion, all processes will converge around here. So let's start. The processes interact with each other, and some of them start to escape from the initial region you see, and the escaped point push other point to the other region using repulsion force, and accordingly, points are diffused over a 3D space. As the process goes on, force stability is gradually lowered until zero. In this way, this method could discover all of the local modes with just one-time parallel run. So the next step is tracking. Performing cell detection at the initial position. So we obtain the initial position of cell trackers at the initial frame. And the purpose of the tracking is to keep attracting the tracker's position to the local mode of the density function, which are changing over time. To track many objects successfully, it is the key to avoid next two types of tracking errors, namely merging and switching. In the case of correct tracking, trackers transit like this, but in the case of merging, Different trackers converge to a same local mode, and in the case of switching, trackers switch their targeting local mode during the tracking. So to prevent trackers from merging and switching, we use the spatiotemporal covariation of the transition of cells. This equation shows a transition model described by Markov random field. In this model, we assume the transition of J's tracker is correlated with the transition of K's trackers. The K's tracker is in the same clique, which is denoted by CJ. Okay? And the noise component for the Gaussian distribution. We use this transition model as a prior distribution. This is the objective function with the proposed method. We maximize this posterior probability with respect to the current position of trackers. And we assume the tracker's position at the previous frame and the kernel density function at the current frame are already known. Then the posterior distribution is proportional to the likelihood and prior distribution. The likelihood generates the image force on trackers for attracting them to the brighter region of the density function. And the strength of the image force is controlled by the temperature parameter gamma of the given distribution. And the prior distribution is here. The difference between the transition of J's tracker and the transition of K's tracker follows a Gaussian distribution. Yeah? And its dispersion is inversely proportional to the sigma-scaled Mahalanobis distance between trackers at the previous frame. This prior distribution generates the constraint force on trackers for enforcing their spatiotemporal covariation. So in this way, we can avoid the switching and merging. These slides show how we construct neighbor sets at each frame. At first, we generate complete graph for trackers at the previous frame. Edge cost of the complete graph is given by a square of distances between trackers. And a tree structure is given as a minimum spanning tree, as shown in red here, from the complete graph. For finding the minimum spanning tree, we use the Poem algorithm. And so this tree has the lowest cost. Okay? And then the minimum spanning tree is assigned to the current frame, and we construct the neighboring sets. This movie shows the tracking result of the proposed method. White squares show the position of trackers at each frame, and white line shows the minimum spanning tree. You can see all trackers keep their correct position throughout the tracking, and switching and merging are almost avoided. We have tested the tracking performance using 3 data sets. As a result, approximately 17 to 19% of trackers could be tracked successfully by the proposed method. For the evaluation, we proposed 2 criteria. One is non-overlapping rate. And this is the rate of non-overlapping trackers that successfully avoided merging. And the other is return rate. We made the test data by joining the original image sequences and time-reversed image sequences like this. And we compare the position of the initial frame position of trackers at initial frame and that of last frame, and count how many trackers could return to the correct initial position. Okay, so details are summarized in the paper. The final step of the proposed method is segmentation. We have now all cell centers at each frame. So the next thing to do is to allocate neighbor box cell to the cell center. This step is also performed based on the kernel density function. Simply stated, we calculate the allocation probability based on the density function and allocate neighbor box cell to the high probability region of the density function. In this way, we obtain this kind of tracking, this kind of segmentation result at each frame. Okay, so let me summarize my presentation. In this paper, we propose an automated tracking method for many moving objects to quantify the neuronal activity of C. elegans. The main contribution of this work is to provide a unified framework for addressing cell detection and multiple cell tracking and segmentation based on the kernel density estimation. The fundamental idea of this work is to use optimization techniques designed for a continuous function for processing this kind of live cell imaging data. We are now planning to apply this idea for other issues on bioimage informatics, such as the non-rigid registration with the free-form objects. And also, the current tracking method cannot deal with the split and merge of cells, but the detection and tracking of cell division is very important for many live cell imaging data. So we plan to extend this method to account for cell division. And the computation cost is very important. In our project, base numbers of imaging data will be generated in the near future. So the decreasing of the computation cost is a pressing issue for us. Okay. So this research is supported by JST CREST program, and I'd like to thank all the members of the project team. Yeah. So thank you for your time.
Thank you very much. Questions? While you're getting up, I can ask one. So this issue of not being able to detect merges and splits,
yes,
it sounds like you have a bit of work to do to deal with that while at the same time incorporating your clever concept of keeping cohorts together, because you will have to modify the cohort concept where cohorts now change over time, right? So that if you allow for merges and splits, at some point the cohort will actually change, right? So do you have any thoughts on this as to how you would do this?
We are thinking about it. It is possible. We think the most simple way to detect graph and division is to perform cell detection for every frame. Okay. Now we detect the position of cell at only at the initial frame, but we can detect for each frame. So this is a simple idea. But of course, I'd like to develop more sophisticated way in the future work. Thank you.
Yes, please.
Why is your assumption of the cell being at the mode a correct assumption?
Can I confirm your point? Your question is
Why is your assumption that the cell is at the mode of the density estimator valid assumption?
sorry.
Can you go back
yeah,
to your
yeah.
maybe third
Your time
slide?
is up, by the way.
I think it was on the third slide or something.
Yeah.
The slide where you make the assumption in the beginning.
Assumption. Okay.
Yeah. My question is,
yeah,
why
here.
is that assumption valid, that each cell lies around a local mode? Why is that valid?
The shapes of cell is basically globular and ellipsoidal. So we can say the local mode, the central position of cell is consistent with the local mode of the continuous function, right?
Okay.
Okay.
Comment on that. I think you actually have to have really nice leading noise data. Otherwise, if you have some rather wider region, you might have multiple peaks for a single cell. So I think the peak assumption could hold maybe in your situation, but others I think is more difficult. I have another question. So with the min span tree, I think this is also very much tailored to the data that you have, right? Why don't you just use k-nearest neighbor or something for local neighborhood? Because if you have more dense tissue, right, I mean, this would pretty much completely break down because the MST would change a lot with rather slight modifications. So I don't see why you need the MST.
Yes. So you're asking why you construct these neighbor sets? Right.
You could just take the k-nearest ones. I think like an ISOMAP or so would be totally fine. I mean, for your situation, the trees are for sure good. But like, you know, if you have more dense tissue, this would be rather arbitrary.
So
Yeah, the question is to use alternative ways of building the cohort graphs rather than MST, because MST would be more variable if you have a dense tissue.
Why do you need a tree? Why not a graph?
Why did tree? Yes, in our data, the formulation of cells is almost linear.
Yeah, in your
Like
data? Yeah,
this.
for sure. Yeah, that's what I said.
Yes.
Okay.
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 All right.
So yeah.
All right, thank you very much.